All right, welcome to podcast on the immune system part 2. We are going to pick up where we left off with the last one. We had been talking about, actually let me go back a little bit further. Okay. Innate immunity versus acquired, and we were talking about the components of the innate immune system, that non-specific general defense. We talked about barriers. We had just started talking about different types of cells, and we had talked about phagocytic cells that eat invaders, and we had talked about non-phagocytic cells that fight infection when it has hidden itself inside of the body's own cells. Okay, and I believe we were just getting ready to talk about proteins. The idea that chemical proteins can act to fight invaders as well. So let's here we go. Okay, there are certain proteins that we've already talked about that fight infection, and lysozyme is one. It is one of the it's a barrier protein, if you will. Let's make sure my hair is not all, that probably is anyway. All right, the lysozyme is in your saliva, your mucus, and tears and things like that, and breaks down bacterial cell walls. Protein complement is the thing we have not talked about yet, and we'll need to, and I'll talk to you about that in a second. And also interferon, and interferon is the chemical that is secreted by infected body cells to signal other cells to get ready, there's an invader in the area, protect yourselves, and also signal natural killer cells to come and attack the body's own cells that have been infected. Now, what we need to focus on here is this protein complement. This is a group of 30 different proteins, and they all do slightly different things, but generally speaking, they act to poke holes in invaders. Okay, so they are floating out in the blood. So let's say this is a blood vessel. Okay, and we've got blood cells, and we've got proteins, and so on, and water out here, and we might have an invader. Here he is, and we don't like him, so let's give him a mean face. Okay, now he might be floating out here in the blood, and this might be a protein, one of the 30 proteins of the protein complement. Well, if he's floating out here and he sees this protein, this protein right now is completely inactive. Okay, I suppose you could think of it too as a zymogen. It's an inactive form. But if this invader swimming, floating past bumps into that, it will become active, and it will act to poke holes in the sky and cause him to explode one way or the other. Again, it's 30 different proteins. There's lots of different things they can do, but the bottom line is they act to destroy invaders. Okay, so it's a general response. If anything bumps into these proteins, they will, that is foreign at any rate, they will have an effect or attempt to have an effect on it. Okay, so a general innate response. Okay, now another part of the innate response that you may have heard of is the inflammatory response. If you've ever experienced inflammation, it's not a real pleasant sensation, but it's an important one. The inflammatory response helps to ramp up, get it going, invigorate this innate immune response we've just been learning about. At the site of a localized injury, at least that's what we're going to focus on. It also has an effect on just general infection from an illness too, and we'll talk about that. All right, the critical cells here are called mast cells. Okay, and if this is your skin surface, all right, in the connective, there's epidermal cells, that just means skin cells, that are immediately at the surface of your skin, okay, making the essential barrier. But underneath it, there's some pretty tough connective tissue. It's not cartilage, but cartilage-like. Cartilage is a type of connective tissue. It's tough, and it helps hold the skin to the muscle and make it durable. Well, in this connective tissue, there are mast cells that are hanging out, and these cells contain sacs of chemicals in them. The chemicals that they contain in their little sacs are called histamines, okay? So when you rip open your skin, we've cut the skin open, or maybe we got a splinter or something, we've opened the skin. Well, when we do that, we generally open up mast cells that might be in the area, and they spill their guts all over the place. And what their guts are is histamine. It's a chemical, okay? Now, histamine has certain effects on nearby tissues, in particular effects on nearby blood vessels, okay? When there are near the skin, or not directly at, but just below the surface, there are blood vessels, okay? Well, histamine reaches these blood vessels and causes the blood vessels to get wider. It causes them to dilate and expand, okay? Get wider. Now, why do we want to do that? Well, we're going to bring extra blood to the site of this injury. So now I'm making my blood vessel wider here. Why would I want to bring blood to the site of the injury? That sounds dangerous. Well, I'm bringing stuff to heal the injury. First of all, I'm bringing platelets and clotting proteins, clotting factors that are going to plug up this hole, for one thing. Another thing I'm bringing are all kinds of white blood cells, okay, to help fight the infection. Additionally, histamines act to make the blood vessel more permeable to fluids, meaning it makes them leaky. And so that means the white blood cells that are contained in here can creep out. They creep out. They look almost like amoeba creeping out, and they are going to come to the site of injury and attempt to destroy any pathogens that have come to the area. Okay? Now, this increased blood flow that we're experiencing, however, brings fluids that flow in. Again, we're trying to get all the stuff to the site of the injury, so it brings fluid. The fluid seeps into the tissue. Extra blood means we're going to increase the temperature. So we're going to cause swelling, which the technical term for swelling is edema, EDMA, and inflammation, which means an increase in temperature. That does not feel good. When you get a cut and it's swollen and it's hard, sensitive to touch, it feels hot, that's the inflammatory response. It doesn't feel good, but it's doing good. It is bringing blood cells and proteins and things to the site of injury. The increase in temperature causes the environment to be inhospitable to any bacteria that might be gaining entry. So it's a very good thing to happen. Now, you might have heard of something called an antihistamine, which you might buy at the store. If you are having an allergic reaction, the mast cells here are releasing their histamine contents for no good reason. They are doing it as a response to something they think is a pathogen, but it is not. And you are having this inflammatory response when you don't need it. For example, if your mast cells sense that pollen is an invader, they will release histamine, and that causes the fluid to come to your eyes, for example, where the infection might be happening, mucus, extra fluid secretions in your nose, and you feel very unhappy. So because of that histamine effect, you would take an antihistamine in an effort to calm down those histamines and make yourself not feel so bad. So, at any rate, that's the inflammatory response. Overall, a good thing. We just don't want it when we don't need it. A more generalized inflammatory response we would recognize as a fever, and histamines play a role in ramping up fever. There's actually other things that do that as well, but that's another type of inflammatory response. Okay, and that's just showing you essentially what we just talked about, the white blood cells leaving the capillaries. These are microbes coming in. The one thing that does not appear to be shown here are the actual mast cells, which would be up in here. All right. Now, that's pretty much it for the innate immune system. Please remember, innate and acquired immunities, as we already know, they are different. Okay, one is general, one is specific. One will give you long-term permanent immunity to something, the other one will not. But they are also very connected. This is an important thing to keep in mind. The innate immune response helps to get this acquired immune response ramped up. It helps to get it going, and we're going to talk about that. Certain phagocytic cells will actually send out chemical signals that will help that acquired immune response to be called to action. We're going to see this word cytokines. That's just the name of the chemical that the phagocytic cells may release to help get that acquired immune response going. Regardless, both of them are absolutely necessary. Okay, for an effective immune response. We're going to get started on acquired immunity, and then we'll take another break, I think. All right, we've already learned about acquired. Now, you do need this. The cells involved in the acquired immune response are called lymphocytes. Lymphocytes, okay? It is just another type of white blood cell or another type of leukocyte. So leukocyte is the big overarching term that takes in all kinds of white blood cells. Lymphocytes are just one type of white blood cell. Neutrophils and all those other phagocytic white blood cells are other types of white blood cells, so that's how those relate to each other. Okay? Now, acquired immunity certainly involves lymphocytes, but to talk about it, we actually need to start with talking about the critters that are infecting your body. Okay? Because acquired immunity, as you'll recall, as we've said many times, is absolutely specific to very particular kinds of microbes. So how is it then that this acquired immune system can tell microbes apart? They can tell them apart from each other and tell them apart from your own body's cells. How can they do that? Okay? It has to do with the ID markers we talked about. ID markers, okay, cell identifying markers on the surfaces of pathogens, of invaders, are referred to as antigens. So antigens are just a type of ID marker that is on the surface of a bad guy. Something that is foreign to your body is called, the ID markers on its surface are called antigens. Okay? So, all cells have unique identification markers protruding from their surfaces, and we've talked about glycoproteins and glycolipids already. Okay? So that must be a glycoprotein or a glycolipid there, and because it's on a bad guy, on a pathogen, we call it an antigen. Okay? Different pathogens have different antigens. Remember, these are proteins and carbohydrates fused together. Shape is absolutely critical to how they work. So every one of them essentially is different because they have a different shape. Okay? Now, lymphocytes. There are two types of lymphocytes. They are called B cells and T cells, and you've probably heard of them. Okay? Both B cells and T cells have unique receptors indicated by these little green molecules on the surface of this B lymphocyte, this B cell. They have unique receptors on their surfaces, and all the receptors on a given B cell will be the same. You can see that all of these have a very particular shape here at the end, here, and it is allowing them to interact with this pathogen. This thing looks like it's probably a virus. And so it's locking in, its identifying antigens are locked into the receptors on this B site. Okay? So these receptors fit with the unique antigens on the surfaces of various pathogens. All right? Now, this is called specificity, when you've got this unique receptor that's going to interact with this unique marker. But it has real implications for how this immune response works. Now, how many pathogens, different kinds of invaders, do you think are trying to get into your body at any point in time during a day? If you said a lot, you said the correct answer. There are thousands, millions of potential invaders that could get into anybody's body if you consider all the bacteria in the world and all the viruses in the world, and hopefully we wouldn't be ever exposed to all of them, but nonetheless, there's a lot. Okay? Now, if we are going to generate cells that are going to specifically respond to each of these different possibilities, each of these different pathogens, how many different types of cells with their different receptors would we have to create? Thousands, millions. There have to be very, very, very many different lymphocytes, each with different receptors prepared to interact with each particular pathogen possibility that is out there. That seems staggering to me to think about that. I would hope it is to you too, that your body has to arm itself for every possibility. So that means that while there is incredible variety in the cells of your acquired immune system with different receptors on all these different cells, because there is such variety, you cannot have enormous quantity of each. You can have a lot of different lymphocytes, but you can only have a few of each, because you can't have a whole army of every single different kind. So that is a kind of a bit of a limitation on this acquired immune response, and why it takes time to ramp it up. We have to have the correct lymphocyte interact with the pathogen that happens to be invading in your body, and then it has to build an army of itself. Okay, so let's look at this. This slide is picture diagram is really pretty good. Let's say this is the antigen that has decided to invade your body today. Okay, now there is a whole bunch of different lymphocytes out there. You can see them, and all of these different lymphocytes, indicated by these guys, have different receptors on their surfaces. All of them. They're different, and they're running around your body looking to make sure there's not an invader that is going to fit with them. If this guy were to bump into this antigen, this invader, he would say, I can't do anything with that. I don't have the receptor for it. If this guy did, he'd say, I can't do anything with that. But if this antigen bumps into this guy who has the correct receptor to fit that antigen, then this guy says, uh-oh, we've got a problem. And he is going to make an army of himself. He is going to begin undergoing mitosis, cell division, to create many, many duplicates of himself. He is going to clone himself. We call this clonal selection, and it is how your acquired immune system works. The one lymphocyte with the right receptors eventually bumps into the invader, the antigen on the invader that is infecting you, and that is the selection part. So it selects, it bumps in, it fits, boom, it's selected, it is picked, it is chosen by the invader itself. Really, it's got the matching receptor for the antigen. And then this guy immediately starts cloning many copies of himself to create a clone army, if you will, to fight this specific pathogen, of which there are no doubt many, many more in your body than just this one. So, this slide is talking about that. Only one of those lymphocytes will have the correct receptor. That lymphocyte will become activated and immediately begin making clones of itself, exact copies of itself by mitosis. This is called clonal selection. You need to know this term and this idea. Okay? All right. Now, there are two basic types of lymphocytes. Like all blood cells, and this goes for red blood cells, all white blood cells, platelets, whatever, they are all born in the bone marrow, in the marrow of your bones, from stem cells. So in the marrow of your bones, there are cells that do not know what they're going to be yet when they grow up. They're immature, and they, depending on what chemical signals they receive or whatever, can go along a couple of different paths, and then they can go along many paths from that. Okay? So, but all of these cells, these blood cells, are born in the bone marrow. There are many, many types of blood cells. They're all born in the bone marrow. Now, the two basic types of lymphocytes, as we already have said, are B cells and T cells. Now, while they are all born in the marrow, B cells and T cells mature in different places. B cells, hence the B, mature in the bone marrow, where they are born. T cells are born in the bone marrow, but they mature in a different place. They mature, become grown-ups, if you will, in the thymus gland, which is a gland kind of in your throat area. Now, B cells and T cells are in charge of two different types of acquired immunity, and we will talk about both of these. One of them, the B cells, are in charge of what we call the humoral response, and the T cells are in charge of cell-mediated response. Essentially, this one uses chemicals to fight invaders, and this one uses cells to fight invaders. Okay? This one, humoral, makes chemicals we call antibodies. They are proteins that act to fight invaders, whereas this one, we already said, the cells are going to do the attacking, not proteins. Okay? Now, this is important right here. These are going to attack foreign invaders that are extracellular. All that means is outside cells. Remember, we said that pathogens could be floating in the body fluids, just hanging out and saying, hello, cell, or some of them are going to be actually hidden inside the body's own cells, and those are a little harder to see because they're hidden. So T cells will attack those that are hidden in the body's own cells. B cells will attack those that are floating free out in the body fluids. Okay? T cells are also going to play an important role in destroying diseased body cells, in other words, cancerous body cells, cells that have gone bad in that way. Okay? All right, I'm going to break it here, and then we're going to talk about the humoral response and the cell-mediated response. Okay? Thanks for watching. See you in the next podcast.